Alright. Your question says figure 1.1 is a diagram of the digestive system. Each letter may be used once, more than once, or not at all. You have to give the letter of the part that is shown in figure 1.1. Hydra, take a minute, okay? Go through all of these one by one and then tell me which, you don't have to write anything down, just like go through all the points by yourself, look at the diagram, and then tell me which letter will be used for each function. Okay.
Okay. I think I know the answers.
All right. Okay.
So the first one where
Mm-hmm.
bile is produced, I think it's the gallbladder.
Okay. I will remember one thing that when we went over the topic, I specified one thing. I said where the bile is produced is different than where the bile is stored. So there are two organs related to bile. One organ is the one that makes bile. One organ is the one that stores the bile. So gallbladder stores the bile. It does not produce the bile. Who makes the bile?
Um, the liver.
Yes, exactly. So
So
that is an important distinction. Okay. Liver will make the bile, the gallbladder just stores it until needed. Okay. So that which letter will that be? That will be letter.
okay.
Perfect. Good. Okay. What about the next one?
That's going to be the stomach. So C.
Correct. Good. And then,
Um, is that the liver again?
yes, this is also liver. Correct. And then, where is maltose digestive?
Um, the small intestines.
Correct. Good. And where does the enzyme trypsin act?
Pancreas.
Yes. Okay. It's saying acts. It's not saying produced. It is produced by the pancreas. You are correct. But where does it act? Where does it do its job?
I'm not sure.
Okay. When the enzyme trypsin is released, where is it released? It
Hmm.
is released into the pancreatic digestive enzymes. Where are they released? Which part of the digestive system? They're not released in the stomach. You are right. If the question was who produces trypsin, you would be absolutely 100% correct. Pancreas is the one that produces trypsin. But they are not saying produces, they're saying where trypsin acts. Where does it do its job?
Oh, so the small intestines.
Yes, small intestine. Correct. Okay. Now, they're saying to you a student investigated the effect of bile on the digestion of fat and milk. Firstly, Harsha, can you tell me what is the role of bile? Why do we need bile? How does bile help in digestion?
Um, it breaks down the fats and fatty acids.
Yes, exactly. It helps in breaking down the fat with a process that is known as emulsification. Okay, so it has a process known as emulsification. You have to know the name of the process in which it breaks down the large lipid particles into smaller lipid particles. So it doesn't actually break down the lipids. Okay, it won't do that. It will simply break down the large lipid particles into smaller lipid particles so that the enzyme will have greater surface area so it can break it down into glycerol and fatty acids. Okay, that's why. Who will do the breaking down? Enzyme, lipase. The enzyme lipase is the one who will break everything down. But what will the bile salts do? The bile salts will break down the large lipid particles into smaller particles. So the enzyme just has an easier time doing it. Okay, so it helps out the enzyme by making the particles smaller. That process is known as emulsification. Okay, so bile will do emulsification, which is breaking down large particles into smaller particles. So it's not a chemical digestion, it's sort of like a mechanical digestion, right? And then when the particles are smaller in size, the enzyme will break down the lipid into fatty acids and glycerol. So they have set up three different test tubes. Test tube A, test tube B, test tube C. Test tube A contained milk and bile. Test tube B contained milk and enzyme lipase. Test tube C contained milk, lipase, and bile. So test tube A only has milk and bile. Test tube A has milk and the enzyme only, lipase, no bile. Test tube C has all three, milk, which has lipid, lipase, and bile. They used an indicator that is pink in alkaline solutions and colorless in acidic solutions. Okay, so they wanted to study the effect of bile on the digestion of fat and milk. What is the experiment? Effect of bile on the digestion of fat and milk. So how good is bile in digesting the fat and milk? So they to test it, they used an indicator. Okay, that indicator will be pink if the solution is alkaline and colorless if the solution is acidic. They added the same volume of indicator to each test tube. Okay, first of all, why do you think, like, let's do a discussion. Why do you think, Hajra, they're adding an indicator? How will indicator help them, figure anything out? Like, how will indicator help them get any results? What does an indicator have to do with acidic or alkaline? Like, which part of this experiment is related to acidic and alkaline? Okay,
Or do
yeah, go on.
they put the indicator to show if the like how strong it will be, like the acid or the alkaline?
Yeah, you're right. But my question to you is, where is the acid or the alkalinity going to be coming from? Like, what will be alkaline that I can test? Here is your hint. It is one of the functions of bile. One of the functions of bile is emulsification. Bile has another function. Do you remember what that function was? I will give you another hint. Bile has a compound in it known as sodium hydrogen carbonate. Sodium hydrogen carbonate is needed for neutralizing the stomach acid. Okay. So if you have bile, that means the solution will be alkaline. Okay. That is why we are using an indicator. Okay. So if the bile is present, that means the solution will be alkaline because bile is alkaline in nature. That is why they're adding the indicator. So the indicator will help them figure out the amount of bile. Okay. Because if bile is present, the solution will be alkaline. If bile is not present, solution is not alkaline. Right? That is making sense?
Yes.
Okay, perfect. So what they're doing is that the student observed and recorded the color of the contents of each test tube at 0 minutes, 20 minutes, and 40 minutes. Table 1.1 shows the results of the investigation. Explain the results for test tubes B and C in table 1.1. You don't have to do for A, okay, because A is the, okay, we will see that later. But we have B and C. So in B, you can see Hajra, at 0 minutes, the color is pink. So pink is alkaline, meaning that a lot of bile is present. Okay, or not a lot of bile is present. Basically, the solution is alkaline. After 20 minutes, it's still pink. After 40 minutes, it finally becomes colorless due to the acid. Okay. Then in C, in test tube C, in which you have milk, and you also have lipase, and you also have bile. Okay. So initially at 0 minutes, the color is pink. At 20 minutes, it turns colorless. And at 40 minutes, it is also colorless. So you have to explain the results for the test tubes B and C. Now, I will give you a small idea of what is expected or what is happening in the question, and then you will answer it. Okay. My idea to you is this. You and I are going to brainstorm together. So initially the color is pink, meaning that milk is actually the one. Okay, milk is giving you a pink color because even in test tube A, when there was no enzyme lipase, the color is still pink. Okay, so the color of milk is pink. Okay, now you can see that the color is changing from pink to colorless. That means that the fat in the milk must be broken down. Since the fat in the milk is being broken down, that's why the color is changing from pink to colorless. Right, Hajra? That point makes sense?
Yes.
Okay. Now, when you just had milk and lipase, if there was no bile present, if you just had milk and you had the enzyme, right? The lipase enzyme breaks down the fat. So if you just have the milk and just the enzyme, it takes almost 40 minutes for the solution to turn colorless. So it takes almost 40 minutes for the uh, fat to be broken down. On the other hand, when you add bile, you have the same milk, you have the same enzyme lipase. But when you add bile to it, in 20 minutes, the fat is broken down and the solution turns colorless. Why does that happen? What does bile do, Hajra, that allows, that causes the fat to be broken down quickly? What's the job of bile?
Um,
We just discussed it. I just discussed it with you in the start of this question. Even if you don't remember the name of the process, that's fine. You can just tell me what it does.
it breaks down fats into like
Yes,
fat, like
yes, small. Yes, it breaks
yeah,
down large lipid particles into smaller
into,
lipid particles, so that the
it's easier to digest.
yes, so the enzyme can break it down easily, right? So so basically what happens, Hajra, that it will break large lipid vacuoles into smaller lipid particles. Smaller particles have a larger surface area, right?
Yes.
Enzymes will be able to break it down quickly. Okay, that's the concept. So that is why when you just have milk and lipase, the process happens. Yes, the lipase does break down the fat, the color does change, but it's not that fast. When you break down the particles, large lipid particles into smaller particles, so that there is greater surface area available. Greater surface area available means what? That the enzyme will be able to work faster, and hence the digestion of fat happens in 20 minutes as opposed to 40 minutes. Right? Is that making
Mhm.
sense?
Yes.
Okay, perfect. Now, so this is the answer you basically have to give over here. Okay, this is for 4 marks. So go ahead, put everything that we discussed together in one answer. Okay, and type it out in the chat.
Okay.
All right, Hajra, I got your answer. Goodness, because the file breaks down bigger, those more will have. Okay, one small correction, Hajra, over here, just add those smaller particles will have a larger surface area. Okay, you have written it. Okay, okay, okay. Okay, perfect. All right, great. All right, let's take a look at our next part of the question. Further on, they're saying, explain the purpose of test tube A in figure 1.1. So in figure 1.1, we have test tube A. Test tube A does not have milk. Sorry, test tube A has milk and bile, but there is no lipase. Why do you think they kept test tube A? Considering that test tube A had no milk, it had no enzyme. Why do you think they kept test tube A over there? Like, what is the purpose? How was the test tube A helping us over here?
Um, to show that bile does not help in breaking down fats.
Yes,
Like,
basically to show, yes, to show that the digestion of the fat happens because of the enzyme lipase, and we're basically using it as a control. Okay, this is a control to allow for comparison of data. So yes, this is to show that bile does not break down the fat, lipase does. Okay, bile will just break it down into smaller particles. Okay, we are not interested in the activity of enzyme. Let's take a look at our next question. Just give me 1 minute. Let me share that with you. Okay, take a look at this, Hajra. This is a question where you have to basically fill in the blanks. Your blanks are enzymes are involved in chemical digestion, which produce small dash molecules that can be absorbed into the blood. Two examples of protease enzymes are pepsin and trypsin. Pepsin is produced by dash and requires acidic conditions. These conditions are created by the release of dash, which provides the optimum pH for pepsin activity and also kills harmful dash. The dash produces trypsin, which breaks down protein into in dash pH conditions. These conditions are created by a substance called dash, which neutralizes the gastric acid and also has an important role in the dash of fats and oils. Okay, take your time, go through all of these blanks one by one, figure out what do you think each blank should have, and then let me know when you're done.
should I write the answer in the chat once I'm done?
Uh, I think it's easier, whatever you find easier. If you think it's easier to write them down, or you can just verbally say them as well. I don't mind.
Okay. For the first one, mm,
Yes. Go on, go on.
I'm not sure what they're asking.
Uh, okay, that's fine. I will tell you. You simply are going to say soluble, okay? Because the problem is that there are large particles like starch, for example. Starch is not soluble. Our body, it's not soluble in water, okay? So that's why our body cannot absorb it because our body is mostly, our blood is all water. So that's why the main reason we have to break it down into smaller particles is not just to obviously decrease the size, but it is also to make them soluble so that they will actually dissolve in the water and be absorbed into our blood. Okay. So over here in the first blank, you have to write down soluble. Our large molecules, like all most of some of the proteins, starch, lipids, they're all insolubles. So the entire reason for breaking them down is to make them soluble so that our body can absorb them.
Okay.
Okay. All right. That making sense?
Yes.
Let me know if you need help with any of the other blanks. Also, if you're unsure about any answer blank in the middle, it's fine. Leave that blank and then just figure out the rest of them.
I think I'm not sure about the
Mm-hmm.
um,
It's okay. Just tell me which one you're sure about, and then I'll go over the ones you're not sure about.
I just know where the pepsin and trypsin is produced, but I'm not sure about the rest of them.
Okay, it's fine. I'll help you out. So pepsin is produced by the
Stomach.
stomach. Okay, now these and requires acidic conditions. These conditions are created by the release of what is present in the stomach that is acidic. What is inside the stomach that is acidic? It's in the name, Hajra, acidic. It's an acid.
Oh, so hydrochloric acid.
Yes, hydrochloric acid or gastric acid or stomach acid, right? Any of those would be correct. So over here, you can write down, gastric acid, for example, or you can write down hydrochloric acid. The which provides the optimum pH for pepsin activity and also kills harmful what? What is harmful that the stomach acid will kill? If you remember, there were a few functions of the stomach acid. One function of the stomach acid was to provide the acidic conditions for the enzyme pepsin to work. Then the enzyme also destroys what? What does it destroy? Harmful.
Um, Bacteria.
Yes, exactly. Harmful bacteria. Correct. Okay. Now, who produces trypsin?
The pancreas.
Correct. The pancreas will produce trypsin, which breaks down protein in dash conditions. So, inside the stomach, we have acidic conditions. Inside the small intestine, we have which conditions? We have the, do you remember? In small intestine, we just discussed it as well in relation to bile. We have alkaline conditions. Okay, so they're opposite to each other. Stomach is acidic and small intestine is alkaline. Okay, so pancreas produces trypsin, which breaks down protein in alkaline conditions. Okay, so the enzymes of the stomach work better in acidic conditions. The enzymes of the small intestine work better in alkaline conditions. These conditions are created by a substance called what? We just discussed it. Last question, Hajra. Our last question, what was producing the alkaline conditions?
Um, was the bile.
Yes, correct. Which neutralizes the gastric juices and also has an important role in the dash of fats and oils. Do you remember that I had told you a process that the bile is involved in a process where it breaks down large fat particles into smaller particles? Do you remember what the name of the process was?
Emulsification.
It's emulsification. So the last blank is emulsification. Okay. All right. Let's take a look at our next question. Give me 1 second. This one was making sense, right, Haja? Like you get all of the blanks.
Yes.
Perfect. Let's take a look at our next question. 1 second. A small intestine is lined with many villi and is adapted to absorb glucose. Okay. Figure 2.1 shows two villi from the lining of small intestine. State the name of the structure N on figure 2.1. Do you, if this is structure N, the one in the middle, can you tell me what do you think the structure N is? If you remember, I taught you the structure of the villi. And in the center of the villi, we had this structure that was responsible for absorbing fats. Do you remember what was the name of that structure?
Um, it's the one in the middle, right?
Yeah, the one in the middle.
The lac lactea.
Lacteal, correct. Good. Cell M in figure 2.1 secretes mucus. State the name of cell M. Do you know what is the name of the cell M?
It's the outer one.
Yeah, the yeah.
The microvilli.
1 second. Let me tell you.
Or is it going to be the capillary network?
No, this is not the capillary network. Capillary network are the blood vessels around the lacteal. Okay, so the
Oh,
blood vessels that will
okay.
be all around the lacteal are the capillary network, and this small hair-like projection, this small, you see the small hair-like projection.
Yeah.
This is the microvilli. This is a cell that is known as goblet cell. Okay, goblet cells are not just found in the stomach; they're also found in your respiratory tract. Goblet cells have the function of producing mucus. See, so that was the next question. State one other place in the body where mucus secreting cells are found. So that would be in your respiratory tract. Okay. You also find them
Mhm.
in the reproductive tract as well. Okay. Now, this Haja I am giving to you as your homework. Describe and explain how the structure of a villus is adapted for its function. Okay, so if you remember in the structure of a villus, you need to talk about microvilli. How do the microvilli help in its function? And if you remember, I actually give you a proper answer, right? Like in the notes, if you will see, I give you a proper answer that these are the adaptations of a villi that make it so good at absorbing food, right? That's the function. The function is absorption. So I told you the features of the microvilli. I told you extensive capillary network, all of that, right?
Or
So you,
yeah.
yeah, so you have to put, you just have to write down the same thing, but write it down in a proper answer format. Okay. So take that
Okay.
as your homework and take your time. You can write it down and then just send it to me on WhatsApp. Try to do it by today if possible. Otherwise, send it to me by tomorrow. Okay.
Okay.
All right. Okay, Hajra, will go ahead and stop it over here for today, and we will continue in our next class.